Right. So as you can see from the title, we're trying to stay as math-free as possible. And part of the reason for that is, I wouldn't consider myself a computational math scientist. I really cannot derive a differential equation for you, but what I can do is try to explain kind of conceptually the foundations for large scale modeling using a virtual brain. And hopefully that's helpful to you. I think a lot of us come from a similar background as I do, which is kind of an experimental psychology or cognitive neuroscience or neuroimaging. And so this is the perspective that I share. And what I'll preface the whole series with is this is probably the perspective all the teachers and all the session leaders, I guess, has. And so, we teach what we can, and then beyond that, we kind of will learn from each other. Right? The goal for today, and we'll see how well we achieve this goal and later on in the discussion session, is that I would hope that everyone here will be able to formulate a research question for a project that uses the Virtual Brain platform, and that they're able to design and plan a simple modeling study. And in order to do that, we have to give you some foundations for what the virtual brain can do, and then allow you to formulate hypotheses you might want to test, and so on and so forth. That's the goal. Just a quick outline for what we're going to go through today. One, we'll talk a little bit about motivation, like why we might use virtual brain for large scale modeling. We'll give you some foundations for dynamics, and specifically resting state dynamics. We'll talk about the role for structure and function, especially as it relates to structural and functional connectivity, and what that means in terms of putting constraints on a kind of a space-time relationship. We'll go over what we call vanilla TVB workflows and models, and what kind of a very typical looking model or workflow would look like. The reason we want to teach this is, if you can get your foundations established in kind of a vanilla type flavor of TVB, then you can launch other projects from there. You can add all sorts of fun features to your projects, like stimulation, modulations, or what have you, but we really want to kind of teach a foundational model to begin with. We'll give you an example of some applications, I think one example today, and then at the end, what I was hoping to assess whether we've kind of reached our goal for today is, one, we'll do an Ask Me Anything session. And two, I'll ask that you come up with a project pitch. And that might be a project you're already working with. I know some of you have like a very clearly outlined TVB project you've implemented. Another could just be like, I have this idea, would that work? And I'd love to hear what some of those ideas are and what we might be tackling over the next few weeks. All right, so to start with, the virtual brain considers, or the framework for the virtual brain considers the brain as a complex system, right? Where we have all these levels of organization, from molecules and synapses all the way up to whole brain systems, where each of those neural components interact both within each level and across those levels of organization. They have their own spatial and temporal scales, but that the behavior of the entire system, whether it's the behaviors we're measuring, cognition and cognitive process we're trying to understand, they cannot be inferred just from the individual components of any single, level of organization. And that, together, it's a highly nonlinear system, right? And so together, the interactions produce the behavior, the cognitive processes that we're interested in. So, our measurement tools do try to capture that collective activity at the whole brain networks, whether that's fMRI or MEG or EEG. But what we don't have from any of those measurement tools alone is like a way to interrogate the underlying mechanism, like what is actually happening at the scale of the cell or molecules and whatnot. So the virtual brain platform is a solution for exactly that. It's for kind of linking those scales and connecting those levels of organization. And Virtual Brain really is a software platform. It's a tool, much like FSL would be or FreeSurf or whatnot. It allows people the ability to carry out large scale models and modeling. It considers large scale network dynamics. It allows us to personalize those models. If we have individual data, we can create individual models, and we'll go through what that might look like in the next little bit. It can be applied to disorders. It can be used to test treatment plans or interventions, and it's also open source. And that's kind of a large part of kind of the culture around Virtual Brain and Virtual Brain Consortium is that we really are a community, and that's also what we're trying to develop here. Is that we welcome contributions, we welcome new knowledge to be added to the project, and so we really hope that that's part of what we can build here as well. All right, so I talked a bit about linking scales, and what we have on the right is what we're after, right? The behavior, the cognition, or the symptoms of some disorder that we're interested in. If we are, if we're all kind of cognitive neuroscientists, let's say, at the macro scale, the whole brain level, we have either behavioral cognitive processes, right? Whether it's attention or memory or whatnot, and how the brain kind of enacts those processes. This is the term I'm looking for. What we're after, we could be after, and what we'll present kind of in our workflows is, or what are the mechanisms at the micro scale that kind of are related to the behavior and cognition and symptoms we're interested in. And what the virtual brain operates at is actually in this middle ground, and what we'll call the mesoscale, right? And we'll talk today about how we take kind of multimodal imaging data to link these, to kind of, and in combination with Virtual Brain to link these scales. If there are any questions, please feel free to jump in, because these are kind of foundational concepts I really want to make sure that everyone is clear on before we move on. I'm going to assume that everyone here is familiar with the resting state dynamics, or the concept of resting state dynamics, whether it's measured by EEG, MEG, or fMRI. Okay. But what I wanted to make sure that we are all kind of understanding is kind of the role of noise and what noise plays in those dynamics. And noise here is not artifactual noise. It's not like a measurement tool noise that we're often talking about when we are trying to clean our data of that noise. We're talking about noise here is referring to something that's more of a feature of the brain. And here is like a very nice demonstration from this review paper. I grabbed this figure from this review paper where there are two different systems, one on the left and one on the right. In the first system, what you have are XYZ, which are 3 state variables in this network. And if you ping the system and you kind of resolve for its dynamics, it explores this, we'll call it a manifold, say, until it reaches this equilibrium point. It comes back to equilibrium. So you're pinging a system, you're giving it, let's see, some energy. This is a trajectory of its dynamics, and it reaches an equilibrium and stops. And that's what we see in here in these 3 state variables, is that, you've got some energy, and then it all kind of peters out to nothing very exciting, right? If you inject noise into that system, which is the one on the right, what happens now if you ping that system is that it explores kind of away from the equilibrium. It's still, as it explores more of this manifold. And that noise, what that allows is kind of more physiologically interesting signal. Is that clear then? If you resting state networks are physiologically interesting, right? We talk a lot about switching dynamics, that there are states, you switch from one state to another, maybe that underlies, cognition, thinking, thoughts, whatever. And so in many ways, like those concepts of what we're trying to go after are related to a highly dynamical system, whether that's activity and states that are switching back and forth and whatnot. The assumption here is that you wouldn't have very interesting processes from a system on the left, right? We ping the system and it reaches one equilibrium state, and okay, but does that make sense? All right. Okay, so noise is a very important part of the resting state dynamics in these network level systems. The other part that I want to make sure that we're very clear on is the contribution of structural connectivity to, sorry, I want to make sure I can see everyone in case you raise your hand. Structural connectivity to dynamics. And what we're showing here is kind of like a cartoon version of a network. And I think a lot of you have already seen this. But from the virtual brain perspective, at least in the models that we'll be reviewing and teaching, is these are network-based models. And so that these are, the way we represent the brain is with these nodes or regions that are all linked by these edges or connections. Okay. And, and the input for any virtual brain model is a structural connectivity matrix. And this input, I would say, it has two parts. It has a weight component, so kind of like the capacity of the fiber tracts that run, as the anatomical wiring of the brain, but it also has the distance component or the track lengths. And together, the dynamics of the network kind of arise from both those components, both the weights and also the transmission delays that are imposed by those track lengths. So in this figure on the right here, what we have is a 38 by 38 region structural connectivity matrix. And if we were to flatten this out on this one panel on the left, it would look something like this, right? It's an adjacency matrix or a structural connectivity matrix that says like these are the connections that are in the brain. But if we were to look at the possibilities of connections over time, what you see is something really different if you lay it out with a time component along the x-axis here. And that's because of these track lengths that impose certain transmission delays that at short or finer timescales, there are certain interactions that can happen. So certain connections are relevant. At coarser timescales, it's different bunch and different at medium timescales as well. So there's this space-time component to the brain, and this is kind of this is a foundational concept for virtual brain models, and that's where the dynamics arise. Lost mine. Yeah. Okay, so I think with those in place, we can now start building our canonical model for in TVB. And this is where terminology is going to be really important, at least for us to be clear on what we mean when we call something a local model, where the virtual brain is a model itself, or your virtual brain model sometimes we call that the global model. The local model really is what we mean by the building or the little building blocks we use for TVB. And these are models that represent a small patch of cortex or subcortex, if you may, but a little patch of brain. So that's, what's denoted here. Our local model is something we choose to represent, this little bit of brain in our large network. What we start with conceptually can be local spiking models, right? And so there are lots of models that instantiate, large excitatory pools of neurons. Those pools are connected to pools of inhibitory neurons. They have coupling between them that represent, the neurotransmitter interactions and so on and so forth. And so these are spiking networks because the model output are raster plots or spikes and whatnot. So that level of detail is generally not needed, we would argue, for these kinds of models we're talking about at the larger scale. So what is done is that these spiking models, where every single neuron is represented, are then reduced, and the reduced, I guess, representation is called a neural mass model. And these masses are essentially, you reduce it to a single representation for an excitatory pool of neurons. So that one representation represents the whole, patch of excitatory neurons in that piece of brain. It can also have an inhibitory pool, a single inhibitory pool, rather than hundreds, thousands, and whatnot, and the couplings in between them. The choice of, so this one here is a Wilson-Cowan model. It's kind of a classic neural mass. There are many different types of neural mass models, and the choice that we make in model will really depend on your research question. And so we have in the virtual brain, I can't remember what the last count was, something like 24, maybe more. So now we have more than 24. We have 34 choices for local models. The choice you make really depends on, I guess, what your research question is. There are two classes of models. One would be phenomenological. Let's say you are only after a phenomena like oscillation. And maybe you don't need the kind of physiological realism that other models offer, right? Maybe you're actually after something very, very specific, like you want to know about what happens if you were to apply an MDA antagonist as an intervention for something. Then you would need to have a model that actually has the representation for those NMDA receptors, for example. And so which model you choose really does depend on the question that you have. Okay. We will demonstrate one model in particular in the series. We've worked with a few of these, probably answer a few questions about some of these other choices, but part of the decision-making process in creating your virtual brain, planning for your virtual brain projects is a big one is the choice of model and what you want to choose. Okay. So once we made that choice in local model, we call this a building block because each patch of brain, let's say we've isolated our brain into 200 regions of interest. Each one of those regions will be represented by one of these local models, and then we link them all together using that structural connectivity matrix that we have, so that now we've got local models at each of these nodes, and they're all connected by the structural connectome. And so this is probably the only piece of math in this whole this whole presentation, and this is the generic TVB equation. What it essentially is states that we'll call this LFPs, or the node activity. So for any one of these nodes or regions, the activity of that region is expressed as the interactions between the local dynamics, so that local neural math model, and the global dynamics, so the larger connectome here. It has this very important noise term at the end. We know that that is important for generating some kind of physiological signals. And it also has this extra term that we won't touch on much here, but allows for stimulation, for example, for adding a stimulus to your simulation, or adding some kind of intervention, or some perturbation to your model. I'll point out a couple of terms here that are important. Other than noise, actually, two terms that are important. The noise term is important, and we'll look into that a little bit more. And some of you are probably familiar with this global coupling, this G term. This is a scaling term that scales how coupled your structural connectome is. Like that it's a global scaling factor. And we'll explore that a bit more in an example later on what the model is. Okay. Oh, Andre? Yep. Sorry, I can't see the chat. Oh, there it is. Okay. Look, what does the extra term N represent in this equation? Right, that's the, let me go back up. That is if you wanted to add a stimulation, perturbation, or something to your simulation. So TVB does allow the injection of a stimulus followed by, the regular, followed by a simulation. It allows like all sorts of patterns that you can inject. You can have, like repeated stimulation. You can control the shape of the stimulus and things like that. And that's all kind of part of this first term. We won't touch on that in the series, at least not at first. We're really going to go after resting state dynamics and simulating this canonical kind of vanilla flavored TVB, which is without that, kind of ignoring that term. I just wanted to cover very quickly what inputs in a full workflow for Virtual Brain will look like. As I mentioned, we all have our favorite pre-processing tools to get clean data. What we'll cover here is MRI, in particular multimodal MRI data as inputs. In our case, a very typical process looks like we have T1 images for the anatomical kind of geometry of the brain. We have diffusion MRI for deriving structural connectivity. And we have functional MRI for deriving resting state dynamics. We throw that into some kind of processing pipeline, and we get what we need out of it is some form of structural connectivity matrix. And so, as I mentioned before, if you do it using diffusion imaging, you'll both get the connection weights and also the track lengths or distance matrices. You'll also get a set of clean, bold time series. This is all ROI based, so that what we have are kind of matching parcellations in both your structure and functional data. And we show an example of, for example, on top of having the ROI time series, we might have some functional connectivity matrices. We might also calculate functional connectivity dynamics. And I'll show you what that means in our example. The input to the virtual brain is just the empirical structural connectivity matrix. Okay? And I think this is where oftentimes there's some confusion as what inputs are. The functional connectivity matrices are used later, and I'll show you what that means. So we went over how those structural connectivity matrices inform the connectomes. We choose our neural mass model for our local models, do some simulations. Those simulations produce node activity, or LFPs. There are models that can transform that LFP into a bold signal or an MEG signal or an EEG signal. In the virtual brain, in this case, we extract bold signal, simulated bold, and we calculate some simulated functional connectivity and functional connectivity dynamics. And then we do some parameter optimization to try to find the best set of parameters that give the best fit to the empirical data that we have. So the functional data act as a optimization target. And whatever feature you want to use, and we can talk about, what you want to fit to. You might want to fit to the functional connectivity, but you might want to fit to functional connectivity dynamics instead. There are other ways of fitting, but you use some feature of the empirical functional data as your optimization target. You perform parameter optimization, and now for every individual in your sample, or what, maybe you only have one simulation, you have the best fit model. You take those parameters and you might do some further analysis and assessment. Like we just talked about model identifiability, validation, reliability. You can take those model parameters and ask, whether they scale with your research question. So let's say you are interested in how, any kind of some kind of neuropsych measure, how that scales with your parameters of interest, or you can interrogate the simulated data further. Okay, so that's a very typical kind of vanilla flavored workflow. Any questions here? Okay. Now we'll just jump right into the examples. I'm probably going to call on Justin and Leanne to support this because this is really their work, and they've done a great amount of work recently and in their projects, and which is great because I now have material for these examples. Brandon also, if you wave, Brandon, so everyone see you. Brandon's also done some really great kind of preliminary work that's followed the same workflow, and I'm really happy to see that this is something that we're able to apply to other projects as well. So in this example, we're using the Montbrial model as the neural mass model, or the local model. The model, each node's dynamics are described by two state variables. One is the average firing rate, R, which is described by this equation down here. And then the other state variable is the membrane potential, V. There are some parameters of interest listed in this table here. We'll focus on the global coupling scaling factor G, which I mentioned before. I think noise variance is another interesting, a variable of interest, a parameter of interest, I should say. And what we'll do is we'll hook that up to the virtual brain. We'll simulate some functional connectivity, and we'll perform some parameter space exploration. And so in this case, there are several ways. I think this is this, Leanne, your example, where you've used several ways to optimize these parameters. And the nice thing about it, and this helps kind of model reliability, is that no matter which optimization target you're using, it generally gives you a very similar set of parameters, and parameter space looks fairly similar. And we can kind of unpack that a bit more later. So what we do is we simulate, these are simulated fMRI time series for I think 200 regions of interest. We use a slotting window approach to calculate functional connectivity at different time windows, so in little snapshots of time. And then you correlate all those functional connectivity matrices and you get a functional connectivity dynamics matrix. So that now instead of a region by region matrix, you get a time by time matrix. So every single one of these entries or cells is a correlation between any two windows of functional connectivity in time. And you can see that some, let's call them states or patterns of functional connectivity emerge over time and they disappear. So when they emerge, the colors are brighter, and then when they disappear, the colors are bluer. And what you do is you pick a range for parameter space exploration, a range of parameter values. In this case, we're interested in noise on the y-axis and that global coupling on the x-axis. And you simulate using the combination of noise and G values. You perform one simulation for each combination. And so I don't know how many there are here. There's like 5 by 20 maybe. And you find, in this case, I think this is FCD, the peak variance of this functional connectivity dynamics. And what you see is that the system is most variable at this spot, at this combination of global coupling and noise. There's some range, right? And that's why we do kind of the space exploration, is that you'll get this map where there's a peak, but that kind of falls off. And then you can do this. This heat map gives you the highest variance, but you can also fit to using different ways. So you can either do the max variance, you can do the max correlation between the empirical data and the simulated data. You can do, some kind of comparison between the dynamics in your empirical data or your simulated data, or some combination, which I think, Leanne, you've also come up with some composite score to kind of take advantage of all of these as target features and come up with a composite score. And eventually what you land on is some optimal combination of parameters that gives you the most, I guess, representative simulation for the data set that you have. Okay, so we'll jump now into kind of an example. And this is an example from our healthy aging project that Justin's a part of, and a number of you have heard of or are part of as well. In this case, I think this is an example from Justin's work where he has used the Montbrio local model to simulate healthy aging brains using a CAM-CAM data set. And the data set is something like 650 individuals between the ages of 18 and 90. And we're creating an individual model, a personalized model for each individual in the sample. Once you've simulated your data and you've optimized your parameters, then for each individual, you have your best model. What do you do with it, right? And I think for most of us, the obvious thing is to inspect your simulations. So you can inspect your fold, you can inspect your functional connectivity, you can inspect functional connectivity dynamics, right? You can, and by inspection, I mean, you can look at them, but you can also ask whether they have the same features as in your empirical data or the features you expect. Something else you might want to do is examine the state variables, the firing rate and the membrane potential. Here, Justin's just plotted some of those for one simulation. For each node, their membrane potential and the firing rate. We inspect, I think we'll go into this in a lot more detail further on in one of the later sessions. We can examine their dynamical behavior. These are 4 different regions or nodes, and these are what we call, I guess, bistability plots. Firing rate is on the x-axis of these plots, membrane potential is on the y. And what this demonstrates is that for at least, I don't know, a couple of these, these are bistable behaviors. There's kind of you can see 2 clusters of activities or states. And you might be interested in what that means, what those dynamics mean, right? Like there's this particular node that is exhibiting 2 different behaviors, one in one cluster and one in another. There are some nodes that exhibit very little behavior in one state, but are often traversing only the single state. And you can also take, for example, your firing rate and calculate some feature of that. So in this case, this is a multi-scale entropy curve that is calculated based on the firing rate, the simulated firing rate of the simulation. Yeah. And then lastly, you can also examine the model parameters. And this is, I think, the most interesting part of these models, is because they act as kind of like a computational microscope, right? We don't have access to this kind of information from just neuroimaging alone, but we can kind of interrogate those parameters using models. In this case, these simulations were scaling, again, the global coupling and the noise in 2 different aging data sets. The one on the left is from the 1,000 Brains data set from Ulick, and the second one is Justin's work on CamCam. What you see here is a finding that as age increases in these kind of cross-sectional data, this global scaling, this global coupling variable parameter increases. And what I like about this is that we've been able to reproduce it in this CamCam data set, despite the fact that it's a completely different neuroimaging data, very different acquisitions, very different processing, very different decisions made along the way to get to the input data for the simulations, and even like different ways of optimizing the models themselves. And yet we have a kind of reproducible finding. Okay, so I would say, a lot of for many years we've been worried about, are these findings are these models sensitive to processing choices, and what would it mean if they were? And I'm really happy to see that maybe they're not. And so that's it for kind of the instructional part. Any questions? I mean, we can stop the recording.